Welcome to this presentation on genetic information, variation, and relationship. Living organisms store genetic information in their DNA. This information controls characteristics and is passed from parents to offspring. In this topic, we will explore DNA, genes, variation, inheritance, and how scientists use genetic information to study relationships between organisms. Specification overview and biological context. The central idea is that biological diversity arises from genetic differences, both within and between species, and that these differences are shaped by processes such as mutation, meiosis, fertilization, and natural selection. Students must understand how DNA stores genetic information, how that information is expressed through protein synthesis, and how variation provides the raw material for evolution. This topic also links molecular biology to ecology by explaining how genetic diversity influences biodiversity and species survival. A key specification requirement is the comparison of DNA in prokaryotic and eukaryotic cells, including the role of histone proteins and chromosome structure. Another major focus is the universality of the genetic code, which provides strong evidence for common ancestry and evolution. The topic further explores how genetic diversity can be measured using differences in DNA base sequences, amino acid sequences, or observable characteristics. Beyond genetics, students must understand classification systems, including the use of phylogenetic trees and the concept of species based on reproductive isolation. Biodiversity is examined quantitatively using species richness and Simpson's index of diversity, both of which are essential for required practical skills and exam questions. Structure of DNA and chromosomes, genes, loci, and the genetic code. DNA is a polymer made up of repeating nucleotide subunits, each consisting of a deoxyribose sugar, a phosphate group, and one of four nitrogenous bases, adenine, thymine, cytosine, or guanine. Nucleotides are joined together by phosphodiester bonds, forming a sugar-phosphate backbone. Two DNA strands run antiparallel and are held together by hydrogen bonds between complementary base pairs, adenine pairs with thymine and cytosine pairs with guanine. This specific base pairing is essential for accurate DNA replication and protein synthesis. In eukaryotic cells, DNA is organized into long, linear molecules associated with histone proteins to form chromosomes. Histones allow DNA to be tightly coiled, enabling large amounts of genetic information to fit inside the nucleus while also playing a role in gene regulation. In contrast, prokaryotic DNA is short, circular, and not associated with histones, reflecting fundamental differences in cellular organization. Mitochondria and chloroplasts contain their own DNA, which is circular and resembles prokaryotic DNA, providing evidence for the endosymbiotic theory. A gene is defined as a sequence of DNA bases that codes for the amino acid sequence of a polypeptide or for a functional RNA, such as tRNA or rRNA. Each gene occupies a fixed position on a DNA molecule known as a locus. The sequence of bases within a gene determines the order of amino acids in a protein, which in turn determines the protein structure and function. The genetic code is read in triplets of bases called codons, with each codon specifying a particular amino acid. The genetic code is universal, meaning the same codons code for the same amino acids in almost all organisms. This universality provides strong evidence for evolution and a common ancestor. The code is also described as degenerate, because more than one codon can code for the same amino acid, which reduces the impact of some mutations. Additionally, the code is non-overlapping, meaning each base is read once as part of a single codon. In eukaryotic cells, not all DNA codes for proteins. There are introns within genes and large amounts of non-coding DNA between genes. These regions were once referred to as junk DNA, but are now known to have regulatory and structural roles. DNA replication, transcription, and translation. DNA replication is the process by which genetic information is copied prior to cell division. In this mechanism, each new DNA molecule contains one original, parental, strand and one newly synthesized strand. Replication begins when the enzyme helicase breaks the hydrogen bonds between complementary base pairs, causing the double helix to unwind and separate into two strands. Each strand then acts as a template. Free nucleotides align with exposed bases through complementary base pairing, ensuring accuracy. DNA polymerase catalyzes the formation of phosphodiester bonds between adjacent nucleotides, forming the new sugar-phosphate backbone. Because of complementary base pairing, the sequence of the new strand is determined by the template strand. The semi-conservative nature of replication was demonstrated by the Mendelssohn-Stall experiment, which used isotopes of nitrogen to show that each daughter molecule contains one original strand. Protein synthesis occurs in two main stages: transcription and translation. Transcription takes place in the nucleus of eukaryotic cells, where the DNA base sequence of a gene is used as a template to produce a complementary messenger RNA, mRNA, molecule. RNA polymerase binds to the gene, separates the DNA strands, and assembles RNA nucleotides using complementary base pairing, with uracil replacing thymine. The resulting mRNA carries the genetic code from the nucleus to the cytoplasm. Translation occurs at ribosomes, which may be free in the cytoplasm or attached to the rough endoplasmic reticulum. During translation, the mRNA sequence is read in codons. Transfer RNA, tRNA, molecules, each carrying a specific amino acid, bind to complementary codons via their anticodons. The ribosome catalyzes the formation of peptide bonds between amino acids, producing a polypeptide chain. The sequence of amino acids determines the protein's final structure and function. Sources of genetic variation, natural selection, and adaptation. Genetic diversity refers to the variety of alleles within a population and is essential for evolution by natural selection. Mutations are random changes in DNA base sequence and can occur spontaneously during DNA replication or as a result of mutagenic agents such as ionizing radiation or certain chemicals. Mutations may be beneficial, neutral, or harmful, depending on their effect on phenotype. Meiosis also generates variation through two key processes: crossing over and independent assortment. Crossing over occurs during prophase I, when homologous chromosomes exchange sections of chromatids, creating new combinations of alleles. Independent assortment results from the random orientation of homologous chromosome pairs at metaphase I, leading to different combinations of maternal and paternal chromosomes in gametes. Fertilization further increases variation because it is a random process involving the fusion of genetically different gametes. Natural selection is the process by which organisms with advantageous alleles are more likely to survive, reproduce, and pass those alleles on to the next generation. For natural selection to occur, there must be genetic variation within a population, environmental selection pressures, and differential reproductive success. Individuals with phenotypes better suited to the environment are more likely to survive and reproduce, leading to an increase in the frequency of beneficial alleles over time. This process results in adaptation, where populations become better suited to their environment. Antibiotic resistance in bacteria, where a mutation conferring resistance allows some bacteria to survive antibiotic treatment and reproduce. Over time, the resistant allele becomes more common. Natural selection can lead to evolutionary change and, eventually, speciation if populations become reproductively isolated. Students should be able to describe changes in allele frequency using graphs and data, and to distinguish between directional, stabilizing, and disruptive selection. Understanding natural selection also helps explain biodiversity and the relationships between organisms. Species, taxonomy, and phylogeny, measuring biodiversity within a community. A species is defined as a group of organisms that can interbreed to produce fertile offspring. Classification is the process of organizing organisms into hierarchical groups based on shared characteristics. Traditional classification systems relied on observable features, but modern taxonomy increasingly uses genetic evidence, such as DNA base sequences and amino acid sequences, to determine evolutionary relationships. Phylogenetic trees are diagrams that show the evolutionary relationships between species, with branch points representing common ancestors. The length of branches can indicate the degree of genetic difference. Biodiversity refers to the variety of living organisms within a particular area and is a key concept in both genetics and ecology. Species richness is simply the number of different species present in a community, but it does not take into account the relative abundance of each species. Simpson's Index of Diversity provides a more quantitative measure by considering both the number of species and the proportion of individuals in each species. The formula, D equals 1 sigma n squared, where n is the number of individuals of a species and n is the total number of individuals, allows comparisons between communities. High biodiversity is generally associated with greater ecosystem stability and resilience. Human activities such as deforestation, pollution, and climate change can reduce biodiversity by causing habitat loss and species extinction. Genetic information stored in DNA controls the characteristics of organisms. Mutations, meiosis, and reproduction create variation, while natural selection drives evolution. By studying DNA, scientists can understand relationships between all living organisms. Thank you for watching.